In 2003, an archaeologist named Savino de Lernia drove a four-wheel vehicle into the remote reaches of southwestern Libya, pushing deep into the Tadrat Akakos Mountains, a place so unforgiving that rainfall can fail to arrive for years at a time. A landscape defined not just by heat, but by absence, where wind scours rock and sand into silence and where, by all expectations, the past should have long since been erased. Yet he was not there to study emptiness, but to search for evidence of a radically different world, the Green Sahara, a period when this vast desert was once a living corridor of water, grass, and life. And on the second day of excavation, at a site known as the Takakori Rock Shelter, his team began removing a thin layer of sand and uncovered something that should not have survived. First, a human jawbone, then ribs, and then something far more startling. Skin, intact, leathery, unmistakably human, preserved in a place where preservation should have been impossible. And what began as a single discovery quickly became two. The remains of two women, both in their 40s, both carefully buried within woven mats, both dating back roughly 7,000 years, their bodies held together not in fragments but as complete forms, their existence suspended across millennia by a set of environmental conditions so precise they bordered on accidental. And at the time, no one fully grasped the magnitude of what had been uncovered, because these were not just mummies, they were records waiting to be read. And it would take 2 decades, multiple research teams, and technological advances that did not yet exist at the moment of discovery, before their full significance could be understood. And when that understanding finally emerged, it did not simply refine an existing narrative, it disrupted it, forcing a reconsideration of how North Africa was populated and, more broadly, how human history itself has been constructed. Because, for most of the 20th century, the prevailing view of the Sahara's past was coherent and widely accepted, a model that framed the region as a bridge rather than a barrier, particularly during the African humid period between roughly 14,500 and 5,000 years ago, when monsoon systems shifted and transformed the desert into a savanna spanning millions of square miles, threaded with rivers and dotted with lakes that supported ecosystems filled with animals now associated with entirely different environments: hippos in water, elephants moving through grasslands, giraffes feeding along tree lines, and alongside them, human populations that appeared, based on archaeological evidence, to move freely across this green expanse, with groups from sub-Saharan Africa traveling northward and populations from the Mediterranean coast moving south, meeting, mixing, and exchanging not only goods, but genes, forming a continuous network of interaction that connected the continent in ways that mirrored the flow of a modern highway system. And this interpretation seemed supported at every level, from the thousands of rock paintings scattered across the Tadrat Akakos depicting daily life, herders guiding cattle, families gathered near water, scenes of hunting and domestic activity, to the pottery unearthed at various Saharan sites that match styles found in the Nile Valley and the Sahel, suggesting contact across vast distances, to the stone tools that appeared to blend technological traditions from multiple regions within the same archaeological layers, creating a picture of cultural convergence that felt both intuitive and empirically grounded. And because this framework aligned so well with geographic logic and the available evidence, it became the standard explanation, taught in universities, embedded in textbooks, and largely unchallenged for decades. Yet beneath its apparent certainty lay a critical absence, a gap that few had attempted to fill, the lack of genetic data. And this absence was not the result of oversight or neglect, but rather a chain of reasonable assumptions reinforcing one another, because ancient DNA research depends heavily on preservation conditions that are almost entirely absent in the Sahara, where extreme heat accelerates the breakdown of genetic material, ultraviolet radiation damages molecular structures, and low humidity contributes to rapid degradation, making the survival of usable DNA highly unlikely, especially when compared to environments like Siberian permafrost, European caves, or high-altitude glaciers where cold and stability protect biological remains for thousands of years. And given these realities, Geneticists concluded that attempting to extract DNA from Saharan remains would be futile, directing their efforts elsewhere, while archaeologists continued to interpret material culture as evidence of population movement, relying on objects rather than biological markers to reconstruct the past, and without genetic data to challenge the prevailing model. The idea of the Green Sahara as a corridor remained intact, untested at its core, until the unique conditions at Tacacori altered the equation, because unlike open desert sites, the rock shelter provided a microenvironment that shielded buried remains from direct sunlight and extreme temperature fluctuations, creating a natural preservation chamber where bodies could desiccate before decomposition took hold, allowing soft tissue to survive and, crucially, enabling fragments of DNA to persist within the densest parts of the skeleton. And it was this rare combination of factors that made the site exceptional, transforming what would have been another set of degraded remains into a source of genetic information spanning thousands of years. And in 2019, researchers succeeded in extracting mitochondrial DNA from the two women, offering a first glimpse into their genetic heritage. Though mitochondrial DNA alone, passed exclusively through the maternal line, provides only a limited view, sufficient for identifying broad relationships, but insufficient for detailed population analysis. Yet even these initial results hinted at something unexpected, suggesting that the maternal lineages did not align with established assumptions about the region, setting the stage for a more comprehensive investigation, which arrived in April 2025, when a team led by paleogeneticist Nada Salem and senior author Johannes Kraus published a study that achieved what had long been considered improbable: the sequencing of full genomes from individuals preserved in one of the harshest environments on Earth, extracting DNA from teeth and dense bone tissue, reconstructing it from degraded fragments through advanced sequencing technologies capable of assembling millions of overlapping pieces into coherent genetic profiles. A process that requires not only technical precision, but also computational power to distinguish authentic ancient signals from contamination and damage. And the results fundamentally altered the narrative, because when these genomes were compared against extensive datasets of both modern and ancient populations from Africa, the Near East, and Southern Europe, the Tacacori women did not fit into any known genetic framework associated with sub-Saharan Africa. Despite their location within a region assumed to facilitate north-south interaction, they showed no measurable genetic contribution from populations south of the Sahara. Instead, belonging to a lineage that had not been previously identified, a so-called ghost population, inferred only through indirect signals in genetic studies, but never directly observed until now. And according to the analysis, this lineage diverged from sub-Saharan African populations approximately 50,000 years ago, around the time when modern humans began dispersing beyond the continent, suggesting that while some groups migrated outward, others remained in North Africa, maintaining genetic continuity over tens of thousands of years in relative isolation, a conclusion reinforced by comparisons with 15,000-year-old remains from Tafaralt Cave in Morocco, which share similar genetic characteristics despite the temporal and geographic distance, indicating long-term stability rather than repeated influx from external populations. And this evidence challenges the core assumption of the Green Sahara model, not by denying the existence of environmental connectivity or cultural exchange, but by demonstrating that such connectivity did not necessarily translate into widespread genetic mixing, implying that ideas, technologies, and practices can move across landscapes independently of large-scale population movement, a distinction that archaeology alone cannot easily capture. And the implications extend further when considering the presence of Neanderthal DNA within the Tacacori genomes, albeit at lower levels than those found in non-African populations, indicating that some degree of contact with Eurasian groups occurred early in their history, followed by prolonged isolation, effectively sealing this lineage off from subsequent genetic exchange. And while the sample size remains small, limited to 2 individuals, the consistency of the data and its alignment with other findings lend it significant weight, contributing to a broader pattern emerging across different regions of the world, where ancient DNA studies reveal populations that once existed but left little or no trace in modern genetic landscapes, as seen in research from the Americas, where analyses of pre-Columbian remains have identified numerous distinct lineages that have since disappeared, suggesting large-scale demographic shifts and losses over time, and similar findings from Colombia indicate complete population replacement in certain areas long before European contact, highlighting the dynamic and often discontinuous nature of human history, a process shaped by migration, isolation, adaptation, and extinction. And these patterns collectively challenge the notion of a simple, linear progression of populations, replacing it with a more complex network of branching, overlapping, and vanishing lineages, where entire groups can persist for millennia and then disappear without leaving direct descendants. And within this context, the Takakori discovery is not an anomaly, but part of a larger reconfiguration of how we understand the human past, emphasizing the importance of integrating genetic data with archaeological and environmental evidence to construct more accurate models, while also acknowledging the limitations and uncertainties inherent in each line of inquiry. Because debates continue regarding the interpretation of these findings, with some researchers cautioning against broad generalizations based on limited samples, and others proposing alternative explanations involving ecological barriers or localized isolation rather than continent-wide patterns. Yet regardless of these discussions, the central insight remains that the history of human populations is far more intricate than previously assumed, shaped by factors that do not always leave visible traces in material culture alone. And perhaps the most compelling aspect of the Takakori story lies not in the conclusions it provides, but in the questions it raises about where this lineage existed during earlier periods, how it adapted to changing climates, and what ultimately led to its disappearance as the Sahara transitioned back into a desert, transforming a once habitable landscape into an environment that could no longer sustain the communities that had thrived there, leaving behind only fragments of their existence preserved under exceptional conditions, waiting to be uncovered and interpreted, reminding us that much of human history remains hidden, not because it never existed, but because the evidence required to reveal it has yet to be found or has not survived, and that each discovery, however small, has the potential to reshape our understanding in ways that extend far beyond its immediate context, challenging assumptions, refining models, and expanding the scope of what we consider possible within the narrative of our species.